Okay, at the top of the hour here, we're very excited to have Han Zhu, PhD. He's an assistant project scientist at UCSD, joining us today. And his talk will be titled Modeling Human Pancreatic B-Cell Development and the Destruction in Type 1 Diabetes. Just a little bit about Han. He currently holds the position of assistant project scientist in Dr. Mikey Sanders' laboratory at the University of California, San Diego. He earned his PhD under the guidance of Dr. Zhenghao Wu at the Hong Kong University of Science and Technology, and his doctoral research focused on delineating the behavior of muscle stem cells during the process of muscle regeneration. In 2018, Dr. Zhu joined the Sander Laboratory as a postdoctoral researcher, where he embarked on research into the formation of pancreatic beta cells and their destruction in context of type 1 diabetes. His work employs advanced high-throughput genomic tools and human stem cell models to elucidate key gene regulatory mechanisms underpinning these processes. Dr. Zhu was a recipient of the California Institute of Regenerative Medicine, or CIRM, postdoctoral training award, and his research has been funded by the DRC, Diabetes Research Connection, which is a very helpful funder to young early career scientists who are innovative in their field. And he's currently actively engaged with the HERN, Human Islet Research Network, as a funded new investigator. So congratulations on that, Han. That's really great. So yeah, take it away. We're very interested to hear more about your work.
Okay, thank you, Monica, for the very nice introduction. And first of all, it's my pleasure to be here and present my work. And also, I need to apologize a little bit because I just got COVID, so my voice is going to be a little bit low, but I'm going to raise it as much as I can. Uh, okay, so I'm going to talk about disease modeling, modeling human pancreatic beta cell development and also their destruction during type 1 diabetes. And so before I start, I want to before I start the discussion, I want to ask one question. So, what do you guys think about what determines the biological features of our body? I know there must be a lot of different answers, but I think one important answer among all these factors is our genetic material encoded in our DNA sequence. So this genetic information will define how we look like through a process called the everybody knows that the central dogma. And there are more than 3 billion base pairs in the human genome, A, T, C, and G, and each one of us has slight difference in the sequence, slight variations in this DNA sequence. So the variations in the protein coding genes will affect the function of a protein, and this will lead to phenotypic outcome. So most of the sequence variations associated with the maturity onset diabetes of the young or MODY, and a subgroup that will affect type 1 diabetes belong to this group. Well, more than 95% of our genome are actually not coding for proteins, and within these non-coding regions, you can usually find DNA elements. We call them cis regulatory elements. So these are basically DNA elements that regulate the gene expression in a spatial temporal order. For example, it might regulate the expression of a certain gene within only one cell type, but not the other one. And on the other hand, during the developmental process, it can also regulate the gene expression in a temporal manner. So a lot of the DNA genetic variations associated with these non-coding regions in type 1 and type 2 diabetes are usually located in the CRE region. However, studying this complicated gene regulatory mechanism during disease pathogenesis and development is very challenging because on the one hand, the detailed cis regulation is not completely understood in the human genome. And secondly, because there is difference in the genome architecture between human and other species, so it's hard to use other model system to study this process. So my research program basically combines high throughput genomic tools and assays and human beta cell models to try to address this challenging issue. So I'm going to use one example, a collaborative work with Dr. Kyle Galton at UC San Diego as an example, and to show you like how this type of research is done. So basically, I think I don't need to introduce too much to this audience about what is type 1 diabetes. So people know that this is we know that type 1 diabetes is an autoimmune disease now. So through a very complicated antigen processing and presentation process, our own immune system start to attack our own beta cells and start to attack our own beta cells. And consistent with this this mechanism, the human genetic the human genome-wide association studies or GWAS studies focusing on the T1D genetic risk also show a very similar result. So what you're looking at here is a Manhattan plot summarizing all the T1D GWAS studies. And each dots here represent a genetic risk, and the higher the genetic risk is in the y-axis means the higher this risk is associated with type 1 diabetes. As you can see here, the genetic variation with the highest T1D association is located in this human leukocyte antigen locus, HLA locus. This further support that this disease is an autoimmune disease. But if you zoom into this background region, you also find other, I'm sorry.
Yeah, don't hesitate to take a sip of water.
Okay, yeah, thank you. Thank you so much.
Sure.
Yeah, so if you look at these other genetic risk variants in the genome, you see there are also a lot of them that can also elevate the likelihood of getting the disease very significantly. And a lot of these variations are located in the non-coding region of the genome. So the question is, how does the genetic risk variance in the non-coding genome contribute to type 1 diabetes pathogenesis? So I know there are multiple different mechanisms, but we have one hypothesis that maybe the T1D genetic risk variance will potentially regulate beta cell fragility, human beta cell fragility in this manner, and then cause the disease. So they will make these beta cells have some intrinsic problems to survive the pro-inflammatory conditions. And so to study this process, our collaborator, Dr. Carl Dalton and his lab has mimicked the pro-inflammatory condition in type 1 diabetes by treating the primary human islets with a cocktail of cytokines, interferon gamma, interleukin beta, and TNF alpha. And through a complicated genomic profile, they were able to identify CREs that are induced by these cytokine cocktails, and they were also able to link these CREs to their downstream target genes through high chip and RNA sequencing processes. And on the other hand, we conducted a genome-wide CRISPR screen to look at like regulators that control beta cell survival under this same cytokine cocktail treatment. So we were able to identify genes that are protecting the beta cells under this condition and genes that are promoting beta cell death during this condition. So integrating these two type of analysis, we can now categorize all the CREs into a protective CRE if they are regulating a protective gene, and a CRE, pro-death CRE if they are regulating a pro-death gene. So T1D genetic risk could potentially attenuate the activity of a protective CRE and then lead to the downregulation of the protective gene causing beta cell damage, or it can also elevate the activity of pro-death CRE, then activate the pro-death genes. So to test which one of these two models is correct, we again perform genetic risk enrichment analysis. And through this analysis, we found most of the T1D genetic risks are only enriched in those protective CRE, supporting that the first model is correct. So at least at this point, we made the hypothesis that the T1D genetic risk may attenuate the activity of protective CRE. However, with the beta cell model at that time we used is the NOC beta 1 cells, we were not able to directly mimic the effect of a genetic risk because these cells do not cannot we cannot generate a disease risk containing cell line with these cells. So there is an urgent need of a better beta cell model to this type of study. And this model actually come very, I know a lot of people already know this. So now we can generate human pancreatic beta cells or even the islet-like structures from human pluripotent stem cell through a 7-stage differentiation protocol. And the final product of this differentiation protocol, excuse me. The final product of the final product of this differentiation protocol is an islet-like organoid structure. You guys can see here, and these islet, stem cell-derived islet or SC islet, they can also sense the elevation of glucose level and secrete, elevate their insulin secretion in this glucose-stimulated insulin secretion assay or GSIS assay. So we think this model is like an ideal model to study the type of study I just mentioned before, because the stem cell model is highly compatible to most of the gene editing tools. Basically, we can generate a cell line, stem cell line that contain the risk variants and see whether they affect the downstream effect, have downstream function effects. In addition to that, it also allows to model the complexity of human islet, because in this model it does not only contain the insulin secreting beta cells, it also has the glucagon secreting alpha cells as well as some statin producing delta cells. And there are also efforts to add in additional cell types into this model, including endothelial cells and potentially immune cells to study potential immune cell beta cell interactions for type 1 diabetes study. But before we can like very confidently use this model to study to do disease modeling, there are a few major hurdles we want to address here. So the first one is that the linear decision of this model, especially the beta cell linear decision, is not very accurately controlled. And there is the production of this SCEC population, the atrochromaffin-like cells, which are thought to be islet aberrant. And the other thing is that these SC beta cells doesn't really resemble human adult beta cells. Actually, it resemble the fetal beta cell more. Means their functionality is lower compared to the real human beta cells in the adulthood. So I want to spend maybe the first few minutes, maybe 10 minutes to talk about the first major hurdle, the production of the SCEC cells. So these SCEC cells or atrochromaffin cells, they were characterized by the expression of high expression of genes involved in serotonin synthesis. And in fact, these cells also can also produce and secrete serotonin. So that's one of the reasons why they're called the SCECs, atrochromaffin-like cells. So we want to understand how these SCECs are specified at the level of transcriptional regulation. And to do that, we have collected single-cell genomic data, both single nucleus attack sequencing data, which characterize the open chromatin region in each individual cells, and also single-cell RNA sequencing data, which captures the entire transcriptome in every single cells. And we did this analysis in the last 4 stages of the differentiation, starting from the initiation of the pancreatic lineage. And through our analysis, we can identify different types of pancreatic progenitors, endocrine progenitors, and major endocrine cell types, including alpha cells, beta cells, delta cells, and importantly, we can also find the presence of SCEC cells. So I know like single cell genomic data is like very high order data. Sometimes it's hard to understand. So we try to summarize our findings in by building a gene regulatory network. So as you can see here, this is basically illustration of the network we're building. It's like a sandwich structure, which we can link all the transcription factors to their downstream target genes through these CREs, these non-coding DNA elements. And the beauty of this
Han, can you go back just for 1 minute to that other slide? Can you just sort of unpack this computationally reconstructed cell? What does that really
oh
mean?
yeah. So basically, because at that time we're collecting the single nucleus attack sequencing data and single cell RNA sequencing data separately. So to build this network, we want to like merge the different type of data into like the same cell or same pseudo cell to
Mhm.
allow us to do this network prediction. So we did that through like a computational integration method. So
Mhm.
basically, we try to match the cells with similar epigenome and transcriptome, and then try to link them together.
Right. And what was the method you used?
Oh, that's a good question. So I think we're at that time we're using the SIRAP method. So it's like we are generating pseudo cells, like a group of cells, maybe I don't remember the exact number. We are generating these pseudo cells and we try to match the identity of the pseudo cells from a TAC-seq data and the RNA-seq data through the CRAT integration method.
Got
And
it.
and then if these like pseudo cells they match in their identity, we can assume that the RNA data from the same pseudo cell and the ATAC data from the same pseudo cell, they are like from the same cell. We can basically merge the data set into like this pseudo cell has both the RNA and epigenome features.
Got it. I just wanted to kind of clarify that. Thank you so much.
Yeah, thank you. Yeah. So I think one beauty of our gene regulatory network analysis is that because everything is centered on the CRE. So basically, if we can assign cell type specificity to the CREs, we can basically subgroup this gene regulatory network to allow us to have cell type specific gene regulatory network. And this is how we did it. What you're looking at here is a CRE UMAP. So it's not it's different from the conventional UMAP in the sense that each dot in this UMAP represent one CRE, not a single cell. Um, and we selected the CRE, we selected the high variable CREs from the dataset and try to let the CREs aggregate by themselves into cell type specific CRE modules. And they do aggregate pretty well, as you can see here. This is a CRE module that is specific to the endocrine progenitor cells. And in the middle here, this is a CRE module that is specific to the beta cells. And here, this is the CRE module that is specific to the EC cells. So we try to look at, or we can also look at what are the transcription factors that are predicted to bind to the CRE regions using motif analysis. And we can basically predict the activity of these transcription factors in a cell type specific manner. And within these CRE modules, you can see there are transcription factors that are only active in the in the SE beta cells, and there are also some CREs that are only active in the SE-EC cells, like those ones. And we also find CREs that are active in the in the endocrine progenitor cells like NKX61, and they keep their activity in both SE-EC and SE beta cells. Uh, for example, this one. So we decided to take a further look at this transcription factor. NKX61 is a very important lineage determining factor for beta cells. So we want to see whether they are how does the downstream target gene look like under in both SEEC and beta cells. After careful examination, we found although NKX61 is positive in both SEEC and SE beta cells, they are regulating different target genes in the different cell types. So this is the overlap of the target genes. You can see there is a very few overlap. And interestingly, in SC beta cells, the NKX61 transcription factor is mainly regulating beta cell identity genes, while in the SCEC cells, they are mainly regulating serotoninogenic synapse genes and other not very relevant gene programs. Another very interesting transcription factor we want to look at is this transcription factor CDX2, because this is one of the earliest transcription factors start its activity in early endocrine progenitor cells and also keeps the activity in the SEEC. But while in the SE beta cells, it kind of lose the CDX2 activity. And again, using our network analysis, we can also predict the downstream target gene of CDX2. and we found CDX2 is regulating the major genes involved in serotonin synthesis, including TPH1 and SLC18A1. So we decided to, so we think maybe CDX2 is a regulator of the SEEC lineage, and so we decided to study its function in the differentiation protocol. To do this, we generated a CDX2 knockout cell line, ES cell line, and differentiated the CDX. Oh, sorry, excuse me. And differentiate this CDX2 knockout ES cell towards the SE islet. As you can see here, we have a very clean knockout in the stem cell model, and we again we perform a single cell RNA sequencing analysis to look at the changes induced by the knockout. As predicted by the gene regulatory network analysis, we do see downregulation of major serotonin synthesis gene in the SEEC after we knock out CDX2. And we think, as we think CDX2 may be like a specifying factor for the EC, we predicted that the SEEC population may shrink after CDX2 deletion. And this is also what we have observed. So the yellow orange population here is the EC population. We do see a reduction of the EC population after CDX2 knockout. However, to our surprise, we also observe like a very significant reduction of these SC beta cell population in the differentiation. And in compensation to that, we saw like an elevation of the SC alpha cell population. So this lead us to the hypothesis that maybe CDX2 is an early factor that regulates both EC and beta cell population, and it's also possible that the SEEC population might be a beta cell precursor cell that if you reduce the SEEC population, you also lead to the reduction of the SE beta cell population. So we made this hypothesis because the serotonin production is also a characteristic of the beta cells. There are stages when human beta cells start to upregulate their serotonin production, especially during pregnancy. So we want to test this hypothesis that do SEECs share similar transcription programs with fetal beta, some of these fetal beta cells. To look at that, we first want to identify whether there are serotonin-producing fetal beta cells during the fetal developmental stages. To do that, we collected human pancreas sections through different developmental stages in the human, in the fetal stage, neonatal stage, infancy stage, and also the childhood stage. Interestingly, we did observe a significant number of insulin-producing beta cells also containing the serotonin production, and this percentage dropped gradually after birth. And they keep some of the infants do keep some of these cells, but they definitely disappeared in the childhood stage. We also to validate whether SEECs are similar to these fetal beta cells at the transcription level, we also collected data, single cell RNA sequencing data from a primary human fetal islet study, and we try to integrate these their data set with our stem cell derived islet data set and compare the SEEC, the fetal beta cells and the SEECs. And through this integration analysis, we see that the fetal beta cells and SEEC, they co-localize in this UMAP, as indicating their similarity. And in addition to that, we also took out all the fetal beta cells here and tried to perform subgrouping analysis. And through this analysis, we do find a subpopulation here in these fetal beta cells. They have expression of TPH1, very little expression of SLC18A1, but these sub beta cell population, we call them EC-like fetal beta cells, they also have high expression of CDX2. And to confirm this finding, we also checked CDX2 protein expression in the human pancreas sections we collected earlier across different developmental stages. And again, we do see CDX2 expressing serotonin positive and insulin positive beta cells in early stages of fetal development, but the CDX2 expression very dramatically drop before birth, and they keep their expression at a very low level after birth. So this is a running model of our finding. So we think there is a CDX2 positive progenitor population in human development, and in the fetal stages, some of these fetal progenitor cells will become beta cells, but they will also have serotonin expression. And the serotonin positive CDX2 positive cells will gradually lose their CDX2 in the neonatal stage and infancy stage. And going into the childhood stage and adult stage, these cells will also lose their serotonin production. So in summary of this first part, we have compared the transcription programs during human fetal development from the endocrine progenitors to fetal beta cells to adult beta cells. We have also compared this process to the differentiation process, the in vitro differentiation process in the culture dish. A very important finding is that the fetal beta cells share similarity with SEECs, and the similarity lies in the CDX2 serotonin production axis. This actually provide a potential to maybe further differentiate this SEEC and correct their lineage to allow them to become a beta cells. And I'm proposing some possibility here. So for example, we can try to fix the NKX61 binding sites in these SEECs because they are not supposed to bind to the correct site as in the beta cell. And also we're also trying to test whether switching off CDX2 at a later stage will make the cells become beta cells. Because if what we have tried is to knock out this factor in the beginning. While if this is like also involved in the lineage specification of both populations, it will lead to the reduction of the beta cell population. But we can potentially switch it off in a later stage and see whether we can turn the ECs to the beta cells. And in addition to this first part, we are also trying to understand why these acid beta cells resemble human fetal beta cells, but not the adult beta cells. So this means that we need to potentially look at the postnatal development of human beta cells with when these beta cells gain maturity during in this developmental process. So this beta cell maturation mainly happens after birth, and there are a few major alterations during this stage. In the beginning, some of the beta cells still keep the proliferative capacity, so there is a increase in beta cell mass. And during this postnatal development, they will gradually improve their insulin secretion, and there is also associated with the metabolic reprogramming process. So to study this postnatal development and beta cell maturation, we use the single cell genomic tools to compare primary human beta cells in the childhood stage, primary human beta cells in the adult stage, and try to compare these transcription profiles with the stem cell derived beta cells in the culture dish. And long story short, we also built a gene regulatory network that summarized all our findings. As you can see, this is again a CREU map, which you can find on the top region. Those are the CRE modules that are only specific to the in vitro culture, and at the bottom, these are the CRE modules that are only specific to the primary beta cell culture, primary beta cells. And in the middle, we can also find some common CRE modules that are shared between the in vitro culture and the primary beta cell culture. A very interesting finding is that most of the in vitro culture specific CRE modules are involved in are regulated by transcription factors, lineage determining transcription factors, which means the lineage is correctly established in these SE beta cells. While what they are missing, what this in vitro culture is missing, is mostly the signal-dependent transcription factors, including progesterone receptors, thyroid hormone, circadian rhythm, and other signal-dependent transcription factors, which are signals that are involved in beta cell maturation but are currently missing in the stem cell culture. Using our gene regulatory network, we can basically summarize all these postnatal environmental signals and potentially look at their downstream target genes and downstream gene ontology programs. As you can see here, we confirmed that the circadian rhythm signal can regulate a lot of genes involved in chromatin modification, indicating that the circadian rhythm and circadian entrainment may trigger a epigenetic memory to sustain their functionality in the adult stage. But we are not very satisfied by just computational prediction. We also want to focus on a specific function that is involved in beta cell maturation and try to look at what are the potential regulators involved in this process. And to do this, we have decided to do a functional directed genetic screen to identify regulators involved in beta cell maturation. And the function we focused on is the glucose-dependent mitochondrial function. As you guys must already know that the mitochondrial function is very important for beta cell insulin secretion, is a key step for the beta cells to sense glucose and secrete insulin. While this glucose-dependent mitochondrial activity is kind of missing in the immature beta cells as well as in the SC beta cells. As is summarized here, you can see in this oxygen consumption rate analysis through seahorse, the real human islet can respond to elevated glucose concentration, while the stem cell-derived islet cannot. So we want to understand what are the blockade that prevent the stem cells from gaining the glucose-dependent mitochondrial function. And to do this, we have conducted a genome-wide CRISPR screen. I should mention here that this work is funded by my pilot research grant from the Diabetes Research Connection, and through this genome-wide CRISPR screen, we were able to, in the stem cell-derived beta cells, we were able to find positive regulators of mitochondrial function in these beta cells and also negative regulators of mitochondrial function. We performed gene ontology analysis. We were seeing like most of the negative regulators are involved in GO terms like fatty acid synthesis. While in these positive regulators, we do see genes related to mitochondria, but we are also seeing cytoplasmic stress granules and other translational initiation factors, which we think that are very interesting. But despite that, we also look back to our single cell. I'm sorry. But we also look back to our single cell genomic data sets and try to understand whether there are transcriptional regulation of beta cell mitochondria activity. So we check our single cell RNA sequencing data in primary beta cells and SC beta cells, focusing on these identified negative and positive regulators. And interestingly, we do see most of these negative regulators have higher expression in the stem cell-derived beta cells compared to the primary cells, and most of these positive regulators have higher expression in the primary beta cells, indicating that the regulation of beta cell mitochondria activity here is under like a transcriptional regulation. So and supporting this idea, we also find a lot of genes involved in histone modification and epigenetic regulation, including EZH2 and NCOA1, are actually regulators of beta cell mitochondria activity in our CRISPR screen. So we think potentially the mitochondrial function in the SC beta cell model is actually repressed through a transcription mechanism. And since we have the gene regulatory network from the single cell data, we were able to predict what are the transcription factors that are involved in this transcriptional repression and transcriptional epigenetic repression program. So we found a lot of genes that are regulating, that are activating beta cell mitochondria activity is downstream of the thyroid hormone receptors. And interestingly, a large number of them, and a large number of these target genes are repressed by NR2F1, another nuclear hormone receptor, in the SC beta cells. So we think there is a counteracting function between these two nuclear receptors, and this may provide a repression mechanism in SC beta cells. So we have now been focusing on like some ongoing work. So we're trying to improve SC beta cell mitochondrial function through manipulating these key transcription programs and see whether we can improve beta cell mitochondria function in the stem cell culture. And we're also investigating what are the changes in acid beta cell epigenome after this manipulation. So this ongoing work is funded by the new investigator Gateway Award from her, and I'm very happy to take part in this process. So in summary of the second part, we use integration of single-cell genomic tools and computational prediction. We found that a lot of maturation-associated signal-dependent transcription programs are absent in the stem cell differentiation culture. And in addition to that, we have also employed a top-down genetic screening approach, and we are now building the hypothesis that the glucose-dependent mitochondrial activity in acid beta cells is potentially repressed by a transcriptional and epigenetic mechanisms. So with that, I would like to thank my collaborators in the Center Lab who are listed here, and also our collaborators at UCSD, at Dr. Carl Galton, Bingren, and also we have been collecting all these human pancreas slides from Vanderbilt and also UK. And here are the support we got from different grant mechanisms, as I highlighted earlier. So part of this second part of my work is supported by Diabetes Research Connection, which provide a lot of help already. Thank you.
Thank you very much, Han, and thanks for soldiering through despite being a little bit under the weather. Appreciate your presentation. It's really excellent
Thank you.
work that's going on there at UCSD, and obviously it's recognized by both the DRC and HERN and others. So I had a question myself, and I wondered, how public is the protocol for beta cell production in vitro coming from sort of the biotech operations? And, in light of this, new paper that you guys put out, in developmental cell that really kind of, you know, came up with the, or, put forth this, this idea that there's insufficient action in the beta cell maturation signals during in vitro differentiation. How might these findings inform in vitro production of beta cells and incorporate sort of the sex hormone signals in titration?
Yeah, I think the protocol is like a lot of so we're now having a lot of different protocols to produce the beta cells in vitro, and most of them are publicly available.
Mm-hmm.
In terms of like what are the ways to further improve this protocol for therapeutic approach, I think like people are also thinking because these cells can mature in vivo after transplantation. So for like the therapeutic approach, maybe that is another option to go to allow these cells mature in vitro. While our focus here is mainly how to use these cells as a disease modeling tool to study type 1 diabetes pathogenesis. So we really want to do this under the in vitro culture. And so this will open up a lot of opportunities for us to perform like high throughput genomic analysis. So that's why we're like focusing on that. In terms of how to improve this protocol, I think this is still a very open question. We have identified several maturation associated signals, and we try to administer that into this in vitro culture. While it give us like a very variable results right now. We think there are still some epigenetic, so these cells are probably still blocked by some epigenetic blocks that doesn't allow them to respond correctly to these maturation signals. So that's why we think it's better to understand like what are these epigenetic features that are still blocking the maturation or like still present in the stem cell culture.
Mm-hmm. Yeah, and then, the work though that you are doing can definitely inform the clinical application eventually, over
Hopefully.
because they can use these findings to refine their not only the the development of the beta cells, but actually even potentially their implantation, correct?
Yeah, hopefully.
Yeah. So it's great. And I would say the Sander lab, Mikey Sander now is in Berlin, right? She's at the Max Delbruck Center. She's a group leader there, I guess, and she's the director of the pediatric uh, group. But you are keeping a robust collaboration with her laboratory, and it seems as if things are working out well, even though they're in two different locations, correct?
Yeah, so yeah, we still keep like try to keep a constant discussion with her. I noticed Matt is also here, so we're still like we're also trying to talk to her relatively frequently to have like scientific discussions and think about new ideas. Yeah, she's still very helpful on that.
That's great. It just goes to show that even geographic, distance, there's still, even with geographic distance, there still can be robust collaboration and significant progress in the work. I got a question here. Great work for EC-like cells in fetal pancreas. Do you have any idea why they are gradually lost in childhood?
Yeah, yeah, that's a very great question. So a short answer is we don't know yet, but we really want to understand that because so we think maybe learning from the real developmental process can further allow us to improve the protocol and potentially provide ways to remove those cells in the in vitro culture.
Hmm, great. Okay, last call for questions. Anyone else? Feel free to reach out to Hanzu. He's located at UCSD and he can answer your questions if any come up via email. And thank you again, Han, for joining us. It was a great
Thank
talk.
you.
Really excellent work that you guys are doing down there in San Diego at UCSD. And we appreciate you joining us today.
Thank
Um,
you. Thank you so much.
and we look forward to seeing what's next for you. Thanks again. Have a great
Thank,
rest of your day.
thank you.
Bye bye.